Hello, my name is Arianna Wood, and I will be presenting on preliminary research and data for a field study on ProBlad Verde, which was conducted in fall 2021. The information in this presentation is still going through analysis and statistical review, so it is subject to slight changes. The objectives of this research were to assess the effectiveness of ProBlad Verde to combat coffee leaf rust, or Humilia vastatrix, and to compare ProBlad Verde to another current coffee leaf rust treatment recommended as an organic control option. I completed the study under the supervision of Dr. Mona Lisa Chatterjee at the University of Southern California and University of Hawaii at Manoa College of Tropical Agriculture and Human Resources, or UHCTAR, Extension Agent Andrea Kawabata. Mention of a trademark or proprietary name does not constitute an endorsement, guarantee, or warranty by the University of Southern California, the University of Hawaii Cooperative Extension Service, or its employees, and does not imply recommendations to the exclusion of other suitable products. With that being said, let's get started. ProBlad Verde was approved by the Hawaii Department of Agriculture under a 24C Special Local Needs label, which will be referred to as the SLN label going forward. This product can be incorporated into a new or existing spray rotation and is most effective when applied before the development of coffee leaf rust, or CLR, or around the onset of disease development. It is an organic biofungicide which works on contact and has translaminar properties, which means that it is absorbed through the leaves. It then attaches to CLR cells and triggers a reaction that eventually causes apoptic death of the CLR spores. ProBlad Verde needs to be applied when it is able to have 2 to 4 hours of drying time, and if a large amount of rain occurs within 12 hours after the product has been sprayed, another spray must occur within 4 days. It is important when spraying to cover the leaves fully, especially the undersides of the leaves, but not to excess. Once a spray application has occurred, do not return to the field for another 4 hours and do not harvest until 1 full day later. Please make sure to follow the product label and the Special Local Needs label and follow the Federal Worker Protection Standards. For more information regarding this product, please visit the ProBlad Verde information page at www.hawaiicoffeeed.com/ProBladVerde, which also has links to the approved product label and SLN label. These links can be found in the bottom left-hand corner of this slide. In this presentation, I will be going over the objectives of this research, the location in which this experiment was conducted, and give a description of the trees selected. Additionally, I will be detailing the different conditions observed and the methods of data collection utilized. I will also review the results and give a summary at the end of this presentation. Lastly, I will discuss next steps for future possible research. The objectives of this research were to assess the effectiveness of ProBlad Verde to combat CLR and to compare ProBlad Verde to another current CLR treatment recommended as an organic control option, Serenade ASO. Other objectives for the research, which will be included in the manuscript submitted for review in an academic journal, will also cover research on diseases similar to CLR and an analysis of CLR's overall impact on the environment and the economy, among other research. In this presentation, we will only be covering ProBlad Verde and what occurred in the field study. The experiment was conducted in Kealakekua, Hawaii at the UHCTAR Kona Research Station. 8 mature Kona typica trees were observed in 4 different conditions, totaling 32 trees, and did not exceed a quarter of an acre. The planting of these trees are 8 foot by 8 foot, which means that on a per acre basis, there would be 680 trees. Using all personal protective equipment recommended, I sprayed each condition utilizing a backpack mist sprayer at a water rate of 70.8 gallons per acre after conducting a sprayer calibration and based on the location's 8 foot by 8 foot planting. It is important to note that this water rate is specific to this field and study. A sprayer calibration should be conducted on a given farm before spraying any pesticide to ensure that all leaves are completely covered and the water rate used is correct for a farm's method of planting, type of sprayer chosen, and other specifications. A website detailing how to conduct a sprayer calibration and calculate pesticide rates has been provided above the photos. While only 8 trees were observed, all surrounding trees were sprayed with their specific treatment to ensure that observed trees were not affected by CLR in the trees next to them. 4 conditions were observed. Condition A was the control, which was sprayed with water. This was done because the other conditions were also sprayed with water along with their given amounts of product. Water is necessary for CLR spores to germinate, and so if the control condition was not sprayed with water, it could have prevented the CLR spores from germinating. Condition B was sprayed with the higher rate recommended by the manufacturer at 20 milliliters per liter. It is important to note that this rate is nearly 4 times higher than the current SLN label rate of ProBlad Verde. Condition C was sprayed with the lower recommended rate for this study of ProBlad Verde at 45.7 fluid ounces per acre. This rate is the product's SLN label rate for ProBlad Verde. Condition D was sprayed with Serenade ASO at its highest product label rate of 4 quarts per acre. Conditions B, C, and D were sprayed with the spreader sticker adjuvant Newfilm P, which was selected because of the rainy weather conditions we were experiencing in 2021. While adjuvants according to the product label are not required, they can be utilized to obtain better spray coverage of the leaves. A physical barrier was placed between conditions B and C in order to prevent product drift, as shown in dark blue. This barrier was taken down after the re-entry interval. The initial data collection occurred on September 29, 2021, which included taking photos of all leaf surfaces selected. The first spray application occurred the next morning on September 30th. 2 weeks after the first spray, the second measurement was taken on October 14th. A second spray application, which was recommended by the ProBlad Verde manufacturer, occurred the next day on October 15th. As per the label, ProBlad Verde can be used back-to-back, but then another product from a different frac fungicide group with another mode of action should be utilized in order to prevent resistance. 2 weeks after the second application occurred, measurement 3 was taken on October 28th. Then, 2 more measurements occurred thereafter every 2 weeks on November 11th and November 24th. Each tree was divided into 4 quadrants: north, east, south, and west. 2 lateral branches were selected per quadrant, and a leaf on each lateral was randomly selected, flagged, and labeled. Photos of the top side and underside of each leaf were taken during each measurement. Overall, 8 leaves per tree were observed during the measurements, and the top side and underside of the leaves were photographed and analyzed. Any leaves that dropped were noted. CLR was assessed in 3 different ways during each measurement. The first assessment was leaf loss. Data was taken during each measurement on which leaves had fallen. The second assessment was the number of CLR lesions or spots on each tagged leaf. Photos of the topside and underside of each leaf were analyzed, and CLR spots were counted and compared over each measurement to determine if new CLR lesions had developed. The third and last method of assessment was percent CLR coverage on the surface of each tagged leaf. Crop leaf photos were analyzed utilizing the smartphone application called Leaf Doctor to determine the percentage of CLR area covering each leaf. The 4 conditions were then compared across the different measurements. During each measurement, I took photos of each tagged leaf. 5 measurements occurred in total, with over 2,000 photos of the leaves being taken. Each photo was cropped around the leaf's surface to remove the background and then analyzed. Data was then collected from this analysis. I am now going to give a further explanation on how the Leaf Doctor smartphone application was used to calculate percent CLR coverage. This application was selected in order to standardize the method of calculating percent CLR coverage on the leaf as much as possible. After taking a photo of a leaf and cropping the background out of the photo, I input the cropped leaf photo into the smartphone application. At this point, I selected colors on the photo that I wanted to measure that indicates the presence of CLR. I then moved the threshold bar at the bottom of the screen to select the area of the color. Leaf Doctor then calculated the percent area of the coverage of the selected colors, which translates to the percent CLR coverage on the leaf. For example, this leaf had about 2.83% of its leaf surface covered by CLR. This graph shows the amount of leaf loss for each condition over the course of 4 measurement dates. The different colors represent the different measurements from measurement 2 to measurement 5. Measurement 1 was not included because there were no leaves lost in the initial assessment. Note that measurement 2 was 2 weeks following the initial measurement and the first application of the products. Measurement 3 is 2 weeks after the second application of the different products. The control had a steady amount of leaf loss throughout the measurements and the greatest amount of leaf loss overall. The high ProBlad Verde condition had the greatest amount of leaf loss in measurement 2, where it then decreased thereafter. This condition had the second highest amount of overall leaf loss. The low ProBlad Verde condition had the lowest amount of leaf loss in measurements 2 and 3, and the lowest amount of leaf loss overall. The Serenade ASO condition had the second lowest amount of leaf loss overall. This is initial research and data. Future research and projects would be able to confirm these findings. This graph shows the average number of CLR lesions for each condition over the course of 5 measurement dates, with the different colors representing the different measurements from measurement 1 to measurement 5. It is important to note that the number of CLR lesions was only counted on leaves retained on the branches. The control condition had a steady decline in number of average CLR lesions. The high ProBlad Verde condition had a high amount of average CLR lesions, which peaked in measurement 2 and then decreased. The low ProBlad Verde condition had a consistently high number of CLR lesions. The Serenade ASO condition had a slight increase in average CLR lesions in measurement 2, where it then decreased with some fluctuations. This graph shows the average percent CLR coverage for each condition over the course of 5 measurement dates. The control condition had a larger increase in percent CLR coverage between measurements 1 and 2, where it then was consistently high and then decreased in measurement 5. This decrease may be due to the control losing 83% of its leaves by this measurement. The high ProBlad Verde condition had an increase in measurements 2 and 3 and then declined. This decrease may also be due to this condition losing almost half of its leaves by measurement 2. The low ProBlad Verde condition had a steady increase in its average percent CLR coverage. The Serenade ASO condition reached the highest average percent CLR coverage of all the conditions in measurement 3. Even though this condition had a lower amount of average CLR lesions, its average percent CLR coverage was still relatively high. Despite the low ProBlad Verde condition having the highest average number of CLR lesions, remember that this condition also had the highest retention of leaves throughout the project. This may indicate that while there were more CLR lesions per leaf and a relatively high percent CLR coverage level, overall, these trees had a greater total amount of healthy photosynthetic leaf surface area compared to the other conditions because more leaves remained on the branches. ProBlad Verde is an organic fungicide with contact kill and translaminar properties. This product provides some continued protection against new spores that land on sprayed coffee leaves. Serenade ASO is an organic fungicide with contact kill properties only. To summarize, we will focus on preliminary data from this field trial of the low ProBlad Verde condition. This condition was sprayed at a rate of 45.7 fluid ounces per acre and is the only rate specified by the SLN for use on coffee in Hawaii. The low ProBlad Verde condition had the highest retention of leaves throughout the project despite having the highest average number of CLR lesions per leaf. This may indicate that while there were more CLR lesions per leaf, these trees had an overall greater amount of healthy, photosynthetic leaf surface area because more leaves remained on the branches. This condition had the lowest average percent CLR coverage, which also may contribute to a greater amount of photosynthetic leaf surface area. It is important for coffee trees to retain as many healthy leaves as possible so that the plants can generate enough energy to support its growth, fruit, and overall health. The price for the low ProBlad Verde condition on a per acre basis is about $37.13 per acre at a rate of 45.7 fluid ounces per acre. In comparison, Pyoxosemium's cost per acre is about $36.65 at a rate of 7.14 fluid ounces per acre. Both ProBlad Verde and Pyoxosemium have translaminar and contact kill properties. In terms of fungicides that have contact kill properties only, Serenade ASO's cost per acre is about $58 at a rate of 4 quarts per acre, and Badge X2 is about $42 per acre at a rate of 3.5 pounds per acre. These prices may change over time. A next step for possible future research is to spray ProBlad Verde prior to or around the onset of disease development and before the rainy season. In Kona, this may be around early spring in the months of February and March. However, this is variable. Another possibility for future research would be to conduct trials on different varieties of coffee and at varying locations, elevations, and during different times of the year depending on local weather patterns. Lastly, future research could include conducting experiments utilizing ProBlad Verde in a spray rotation with other products such as bacillus-based products like double nickel and Serenade, copper-based products, oxidate, or pre-ACRE. For more information regarding this product, please visit the ProBlad Verde information page at www.hawaiicoffeeed.com/ProBladVerde. Thank you very much for attending the presentation today. I want to acknowledge my advisors, Dr. Mona Lisa Chatterjee and Andre Kawabata, as well as the collaborators listed on this slide. Thank you again.